Topologically, the lumen of the alimentary canal is an enclosed exterior tunnel running entirely through the body. It communicates directly with the outside world, from the oral cavity to the anus. Extracellular digestion requires the body to deploy aggressive chemical agents, hydrochloric acid and specialized proteolytic enzymes, directly into this void to break down complex macromolecules. These chemical agents present a significant physiological hazard. The same mechanisms that hydrolyze dietary protein will readily dissolve the organism's own mucosal lining. Survival depends on strictly isolating these violent chemical reactions from the vulnerable, living cells that constitute the body's interior. The digestive tract is a schematic of isolated bioreactors in a precise sequence. Sphincters act as physical airlocks, maintaining each compartment's integrity. This isolation maintains specific pH gradients. The stomach sustains pH 2, while the adjacent duodenum operates at 8.5. Autonomic neuroendocrine feedback loops actively monitor these boundaries, adjusting secretions and motility based on the chemical composition of the incoming food. Digestion is a sequence of highly regulated thermodynamic environments, each optimized for a specific stage of molecular breakdown. The stomach is the primary acidic bioreactor, maintaining a pH of 2. To prevent autodigestion, mucus cells lay down a bicarbonate-rich barrier, shielding the muscular wall. Deeper parietal cells secrete hydrochloric acid to drive this pH, plus intrinsic factor for B12 absorption. Alongside these cells, chief cells synthesize and release the protein pepsinogen. Pepsinogen is a zymogen, an inactive precursor protein that requires a specific environmental trigger to achieve its catalytic form. When pepsinogen contacts the hydrogen ions in the stomach, the acid cleaves the protein, allowing it to fold into its active form, pepsin. Pepsin is uniquely optimized to hydrolyze peptide bonds near aromatic amino acids at this extreme low pH, where most cellular proteins would simply denature. The stomach solves the problem of protein digestion by separating the synthesis of an enzyme from the trigger that makes it dangerous. When the pyloric sphincter opens, highly acidic chyme enters the duodenum, a segment of the small intestine that lacks the stomach's reinforced mucus lining. The physical presence of this acid triggers the release of secretin, a peptide hormone that enters the bloodstream to coordinate a systemic response. Secretin halts stomach motility, signaling the pancreas to intervene. The pancreas dumps bicarbonate-rich fluid into the tract, rapidly shifting the duodenal environment to an alkaline pH of 8.5. As amino acids and fats enter, the duodenum secretes CCK, a secondary sensor. CCK stimulates the gallbladder to contract and prompts the pancreas to release its specific digestive enzymes into the alkaline environment. The duodenum actively and rapidly re-engineers its local environment using these peptide hormones to support the next phase of chemical cleavage. The pancreatic payload contains an arsenal of proteolytic enzymes, including the enzyme trypsinogen, the precursor chymotrypsinogen, and inactive pro-carboxypeptidases. They all arrive in their dormant Zymogen forms. The duodenum dictates activation timing through enteropeptidase, an enzyme anchored to its luminal surface. Enteropeptidase cleaves trypsinogen into active trypsin. This trypsin then catalyzes the remaining Zymogens into their active forms, initiating a localized cascade of protein digestion. By centralizing the activation sequence within the duodenum, the body ensures the pancreas does not process its own tissues during enzyme synthesis and transport. Lipids present a thermodynamic challenge. Being hydrophobic, they spontaneously aggregate into massive droplets that are inaccessible to the water-soluble enzymes in the chyme. In response to CCK, the gallbladder contracts and releases bile into the duodenum to physically intercept and break apart these massive lipid clusters. Bile consists of amphipathic salts that act as a biological detergent, executing the mechanical breakdown of fat rather than chemical cleavage. This emulsification process creates tiny spheres called micelles, which provide a massive increase in the surface area to volume ratio of the lipids. This increased surface area allows pancreatic lipase to access and chemically hydrolyze the lipid ester bonds. For molecules that defy the body's aqueous baseline, physical engineering must precede chemical digestion. As material moves from the duodenum into the jejunum and ileum, the system transitions from chemical cleavage to systemic transport. The architecture of the intestinal villus provides the structural fork for absorbed nutrients, housing both a capillary mesh and a central lymphatic channel called a lacteal. Microvilli line the surface of these structures, providing the massive surface area required for efficient cellular transport. Hydrophilic molecules, like monosaccharides and amino acids, follow the water-soluble route. They use secondary active transport and facilitated diffusion to cross the epithelial boundary into the capillary bed. This network drains into the hepatic portal vein, routing fuels directly to the liver for detoxification. The liver serves as the primary gatekeeper, receiving all water-soluble nutrients before they reach the systemic circulation. Digested lipids face a polarity mismatch with the aqueous bloodstream, necessitating an alternative transport pathway. Long-chain fatty acids and cholesterol diffuse into mucosal cells, immediately reasterifying into triglycerides. These hydrophobic molecules are packaged into large spherical transport structures called chylomicrons. Because chylomicrons are too large for capillary beds, they route into the central lacteal of the villus. This creates a lymphatic bypass. The lipids travel through the lymphatic system, avoiding the liver entirely, and enter the venous circulation at the thoracic duct in the neck. The digestive system manages external matter by compartmentalizing its chemistry, using precise activation cascades, and segregating its metabolic fuel based on fundamental thermodynamic properties.